Hello. It looks like there's a lot of people coming in. I'm just going to give it a couple of minutes. Um, People are popping in like crazy. Probably turn my phone ringer off. Yep. Going to give it a couple more minutes. Okay, so I think we'll go ahead and get started, and people can just pop in as they they get to it. Thanks everyone for joining. I'm Amanda. This is my husband David. For those of you guys that don't know me, um, my husband David was diagnosed with appendix cancer, goblet cell, stage 3B, in January of last year. He had a right hemicolectomy by an appendix cancer specialist, a surgeon who specializes in appendix cancer, and after that, we were told 6 months of chemo every 2 weeks, FOLFOX. So when we said, well, why? They basically said, well, we just don't, we treat it like colon cancer, 50% if you do it, 50% if you don't. That didn't sit extremely well with us. So we started getting a bunch of opinions. And that's how I found Dr. Shen. So Dr. Shen had some data around appendix cancer and systematic chemo, and so he's our doctor, and that's why he's here, because I've asked him if he would come and speak to our group at Appendicure and other really the whole appendix cancer community. So we also have Tim Stuhlmiller, who is VP of Scientific Research at xCures. So I want to thank both of these guys for joining us. We really, really appreciate it. We've got a lot of questions and can't wait to hear what you guys have to say. So with that, I will turn it over to Dr. Shen. And Dr. Shen, I'll share my screen. So you guys let me know when you want me to change the slides. I'm like Vanna.
Okay. Awesome. Yeah, okay, beautiful. We're seeing the slicing.
Let me put this into presenter mode.
Awesome. Well, welcome everyone. Thanks for joining. As Amanda mentioned, I'm Dr. Shen. I'm a medical oncologist, so I'm the type of doctor that specializes in chemotherapy for cancer treatment. I'm kind of unique in that I've decided to focus my practice particularly on appendiceal cancer, and basically all the new patients that I see now have appendix cancer. We started working on appendix cancer almost 10 years ago when I was a fellow at UC San Diego. And back then we discovered that at a molecular level, appendix cancer is very different than colon cancer, but the treatment hadn't really updated to reflect the fact that appendix cancer actually is quite different than colon cancer. So that's really kind of been our mission, is to figure out, learn more about appendix cancer, learn, when can we apply the colon cancer playbook to appendix cancer, and when should we not apply that playbook to appendix cancer. And so we do a mix of clinical trials. We have a wet lab where we put appendix tumors into mice and test new drugs. But a big part of this now is actually trying to learn from all the patients that we treat. And so that overall idea is called real world evidence. Even at MD Anderson, where our main strength is clinical trials, less than 30% of our adult cancer patients will ever go on a clinical trial where you get a drug. Like, we have lots of patients who are on like a retrospective study where we just kind of collect data, but less than a third of our patients will ever be on a traditional clinical trial. And we wanted to do better at learning from the patients who aren't on clinical trials, in part because in appendix cancer, there's only been 8 prospective studies of chemotherapy in appendix cancer, 2 of which are randomized. And that was as of 2022. It was a very nice review article that's listed there. And then we've, since I've been at MD Anderson, myself and colleagues have conducted 4 clinical trials. Three are completed. One is still enrolling. But if you add that all together, that's, in the history of time, less than 250 patients with appendix cancer treated on clinical trials, which is not very many, especially when you consider how variable one appendix tumor is to the next. We know that the subtype matters, whether it's goblet cell or mucinous or colonic type, high grade or low grade. These are all really important differences. And if you mix them together, you're not really going to understand. You're not going to be able to learn anything because it's not going to generalize. And so, next slide, please.
Oh,
yeah, events.
sorry.
Yeah. So, how we're doing this is not super important. We have a collaboration with the subsidiary of the company Palantir. But basically, we've got a way that every patient that comes into MD Anderson, their data is going into a cloud-based system, and we can basically identify, as we see new patients, do they have colon cancer, appendix cancer, pancreas cancer, and from that we've built a very large cohort. You can see colon cancer, which is much more common than appendix cancer. We've got almost 60,000 patients, but in appendix cancer we've got over 3,500 patients, which is quite a lot. And we now get some molecular testing on virtually every patient that we see. Clinical NGS means kind of the standard report that you would order. And then in the research setting, we're getting whole exome sequencing and RNA sequencing as well. And so we hope that we can put all this information together to start to understand, why does a drug work for some patients and not for other patients, by combining the clinical results with the molecular information about the tumor. Next slide, please. And so this is going to show kind of one example of real example of what we're doing. I'm a big proponent of using the drug Avastin, also called bevacizumab. I think it's an important part of appendiceal cancer treatment, but that's really just kind of based on my personal experience as one physician. And that's not really that convincing when you want to convince other doctors to do this. And we need to learn ourselves, who are the patients that are really benefiting from this and who's not benefiting from this. And so we said, okay, let's look at all the patients that we have who's been treated with this drug and see, did it improve the outcome? And so we started with all the patients in our database with appendix cancer. But, one of the major issues is that if you get your chemotherapy at MD Anderson, we have a very detailed record of that from our pharmacy. But if you come to see us and you live in Alabama and, which is 6 hours away, you're not going to drive here every 2 weeks to get your chemo. You're going to get it from a local oncologist. But we don't really have a very good record of that because, the only place that exists in our data is, me typing out, we recommend FOLFIRI and Avastin, but that could be written many ways by different physicians. I don't see all the appendix cancer patients. And so we had to really limit to just the patients who were getting their chemo at MD Anderson. You can see that we went from 3,500 patients to only 600 patients, and then we further had to filter if we were missing other records like the stage, etc. So ultimately, starting with 3,000 patients, we ended up with only 360 that we could evaluate who were treated with Avastin. Next slide, please. And so, we're working on this was presented at the Advanced Cancer Therapies conference. It's a great appendix cancer conference put on by the Society of Surgical Oncology. And, we see it looks like that the Avastin, or also called bevacizumab, is helping patients that, the patients who got Avastin with their frontline chemo are doing better than the patients that didn't get Avastin. But this is a retrospective study, so it's hard to know, why did the patients that didn't get Avastin not get Avastin? Is it because they were already sicker than the patients that did? I generally start with FOLFIRI and bevacizumab in mucinous tumors based on my personal experience, but it's only 17 patients, and so is it because mucinous tumors do better than non-mucinous tumors? In a retrospective study, you really need more patients than just 170 to answer these questions when you actually have to slice and dice all these different possibilities. And so while we're definitely working with the data we have. If we had more complete data and more patients, it would make this task much easier, and we could get to really finer level of precision with who's going to benefit from this drug, which is ultimately what we need to learn. I think that's my last slide.
So this is my slide. Um, before we get to the Q&A session, I want to let Tim introduce himself and xCures. Um, but so our data, my husband's data, our data, I think it's mine, is at a few hospitals. And kind of the point of this slide is just to say, like we're very siloed with our data. It's all over the place, and it needs to be in one central place because we do have rare cancer here. So we don't have tons of data to further the research. So if we could get all of our data into one spot, it could, as Dr. Shen said with his study just now, I mean, it could really make a huge difference in the advancement of treatments and cures for appendix cancer. So with that, I will turn it over to Tim.
Thank you very much for the offer to tell you about what we've been doing. First, myself, I'm a PhD in molecular biology. I have a background in genomics, targeted therapies for cancer, systems biology, really taking large data sets and trying to make sense out of them. Again, for the purposes of cancer treatment, that's been my academic background. And I also have some clinical research, clinical development, early drug development experience as well. And so really just briefly about xCures as a company, we're a startup, a health technology company, and we were spun out from a nonprofit called Cancer Commons, which basically supports individual cancer patients, really usually advanced, stage 4, who are off of standard of care. There's no more options left. And the goal is basically to understand everything that you can about that patient so that you can support them in identifying clinical trials, off-label options, other things that could be done. And so really in trying to support them and obviously nonprofits, others, physicians, researchers, we really needed to put in place a method to be able to collect all the data throughout the country. Sorry, hang on.
While he's dealing with children, and we've all been there. Uh, as far as the Q&A session goes, Dr. Shen has answered all of your questions, but we probably won't get to every single one today. So after this is over, I'm going to send out the entirety of all the questions with all of his answers. So.
Sorry, despite my best efforts, my kids still busted in the room. They just got home from school. So what we did was we really wrapped all this around a observational research protocol. And so every patient that works with us, they have the opportunity to basically donate their data to research. And what this is a national, totally decentralized, central IRB approved research protocol. And patients can sign an e-consent and a HIPAA release. And that basically allows us to go to all the different sites of care and pull all of their medical data together into one place. And we put this in basically a multi-institutional style study so that we can just add other collaborators, really smart folks like Dr. Shen, who can then, utilize that data in an anonymized way to understand, what are the treatment trends? Are there patients that are receiving Avastin throughout the country in all different sites? To be able to answer some of these research questions they might have, just following the standard practice of medicine. And so, myself, as well as a lot of other folks at our company, they have a background in clinical research and oncology trials. And so what we did in building this whole platform was, as we're bringing all these medical records in, we want to be able to create research quality data, but keep the connection back to that source document so that it makes it really easy to verify every data element that it's accurate and it was extracted properly. And so that's really the system that we've built from the very beginning. And we've been a company for about 7 years now. And there's been a real technological advance really in the past 3 years. And that's been what's called the health information exchanges coming online. And this is a result of the 21st Century Cures Act that was put in place, almost a decade ago now. But it's really basically saying patients must have electronic access to their medical records. And you've probably seen this through, most institutions have like a MyChart or something similar like that. But even other institutions now, smaller practices are now like creating these patient portals where you can log in. But still, all of these portals are all siloed. And so the health information exchange basically allows, with a proper connection and proper consent reasoning, right? You can pull down all of the EMR data across all the different systems. And so if you go to the next slide, Amanda. This is kind of what our platform looks like today. We've really evolved it into a full software platform, taking advantage of the health information exchanges, but also all the recent advances in AI and machine learning that have really come across. And so if you just walk across here, a patient can work with us. A lot of patients consent directly to xCelsior, that observational study. But we can also work with other entities, covered entities, institutions, et cetera. And so what we're going to do is pull this data down. We're going to organize all of the medical records, deduplicate them, extract all the metadata so we understand this is a path report, this is a genomics report, this is a radiology report, et cetera. And we do some automated data structuring from that. A lot of this data is already coded, like conditions and labs and vitals like that. But there's a lot of really other critical oncology related data, like what was the histology of the tumor and what was the stage and grade that this biopsy showed on this particular day, or what were all these different medications that the patient received and what were the changes in doses. All of that isn't necessarily coded into the EMR, into the medical records, really consistently across all different sites. A lot of it's just in the narrative of the clinic note. And so what we've done is we've used a lot of different, again, advanced large language models, natural language processing, a lot of AI, basically, to be able to extract all this data at scale. And what we're creating then is this huge catalog of metadata, clinical data that we can then search across and identify cohorts, identify trends and patterns. And then at the end of the day, like I said before, the platform is really built with research so that we can do manual data verification of all these points as needed, just to, again, lend confidence to the data that we're creating at the end of the day. And so, even though we've built this sort of much, much more sophisticated software platform, we've still kind of kept all of this, the mission behind the company of basically pulling all this data together and sharing it with, really smart, dedicated clinician researchers so that they can use it to answer their research questions. I can skip over this pretty quickly. This is just kind of laying out the different data elements that we pull. The stuff on the left is coming direct out of the medical records, and the stuff on the right is some of those algorithms that we've built to be able to extract all this data. And again, this just allows us to scale what we're doing from 1 or 2 patients a day to as many as we could possibly pull on the platform. So I'll show just a few representative examples of kind of what we've done so far. If you could go to the next slide, Amanda. We've had a recent partnership with CPATH. They're associated with the FDA. And this is just an example kind of flow chart of how we worked through with them on sarcomas. It was a sarcoma drug repurposing project. Basically, we worked with a handful of clinician researchers and advocacy groups and identified some patients with advanced sarcoma. They consented to our observational study, and then we went and pulled the data together and created a sort of standard data model for them. And they were really interested in looking at drug repurposing for sarcomas, understanding like what are doctors using just in the real world off label, because sarcoma, it's really just surgery and radiation. That's all they have. And so it's really trying to understand, what are doctors doing? Is anything working? And so the nice byproduct of this is, we were able to sort of create this real world data lake, and then clinician researchers, they can collaborate with us on that to identify, you know, is there some signal here that we should design a study around? Like, can we research on this data and can we just understand, what are we seeing? Next slide, Amanda. This is another example relationship. So we're working with a rare liver cancer advocacy group called Fiber Fighters. They've actually pulled together an international tumor board with experts from like Australia and Europe and all over the US. And basically, there's probably about 200 cases per year. And so they basically offer this as a free service to any patient with this cancer, access to these national experts, international experts. We've been able to publish a few ASCO abstracts on this, and we maintain a public dashboard just to kind of show all the different data that we're pulling together for this effort. This is the last one I'll show. And this is, again, another rare cancer. So this is a rare pediatric adolescent brain cancer called DIPG or DMG. And we've actually built out a natural history study to understand the natural course of disease. And so the big problem right now with getting drugs approved in this space is that there's really no standard of care and there's no natural history. And so there's really no way to understand if a drug is going to work or not. You have to do a randomized trial. And as you can imagine, randomized trials in children that are 5 years old doesn't go over very well with parents. And so, we're trying to pull together this real world data set that has, full source verification, it's regulatory grade, so that we can use that as a benchmark, essentially, for new therapies that are going to be developed in that space. And this is just an example of some of the research that we've done coming out of that partnership, as well as others. And this is just, again, there's a poster that I presented a couple of years ago, and it really just kind of showcases all the different analyses that we can do on this data once we get, once we're able to actually collect the data from a number of different patients. This is an article that we published last year, and this was looking at glioblastoma. So we have a number of partnerships in the glioblastoma space, brain cancer. And this was looking at patients who are receiving proton pump inhibitors and just showing that, particularly for glioblastoma with the specific standard of care drug that they received, temozolomide, that the proton pump inhibitors can actually upregulate one of the enzymes that is basically responsible for metabolizing temozolomide. And so by taking these, over-the-counter drugs that are pretty commonly prescribed, it's actually very detrimental to patients with this cancer. So this is just another example of, what's coming out of our database when we can work with smart physicians that have, good research hypotheses. So that's it for my slides. I just want to thank you all for listening, hearing a little bit about kind of what we're doing. Anyone in the country can participate, and you're really just donating your data to research. And we'll work with Dr. Shen and other national experts to analyze the data and to help answer their research questions.
Yeah, and
all this data is totally free to them. There's no cost. We maintain our company through software licenses to other businesses and other commercial diagnostic companies. It's totally independent from, our mission in supporting patients and research.
thank you, Tim. Thank you so much. And I would love to in a year or so see a case study that you've done in regards to appendix cancer. That would be amazing. And I think that probably everyone wants to help find new treatments and cures. And with that being said, I lost my earbud. So with that being said, anybody who wants help that wants to do this, reach out to me directly. I will be happy to walk you through it. We've already got my husband's data there, and several others who have reached out about it already. So, again, would love to help in any way in that regard. And thanks again, Tim. So with that, we're going to get into the Q&A session. We really only have an hour. I actually scheduled this for 2 hours in the Zoom just in case we went over. But it's really from 4 o'clock to 5 o'clock East Coast time. So we've got another 35 minutes, and we've got all the questions kind of listed out, and Dr. Shen will go through them and answer them one by one. And those that we don't get to, again, I'll send you the full list of question and answers to everyone on this call.
Sure. Amanda, do you want to like read the question and then I'll answer it, or how do you want to?
So, given the rarity of appendix cancer, what key insights have been gained from existing research, and how do treatment responses differ from high grade and low grade disease?
But yes, this is it's a great question. We could say a lot of things about it. I think the key things that we recognize now is that at a molecular level, appendix cancer is very different than colon cancer. We don't 100% know, what that should mean in terms of how to treat appendix cancer patients. But again, that's exactly why we're trying to work with people like Tim and xCures to kind of build the patient outcome databases that will basically tell us this. And also, running clinical trials as well. But like I said, there's challenge in doing prospective clinical studies, and so kind of learning from patient experience data is going to be a little bit faster. The most important feature in an appendix cancer is, in terms of like how to manage it and what the outcome is going to be, is the grade. That's very different than colon cancer, where colon cancer, almost all tumors are moderately differentiated, which is grade 2. Appendix cancer, it runs the gamut from well differentiated, which is grade 1, to moderate, grade 2, to poorly differentiated, it's grade 3. What that means basically is how abnormal does the tumor look under the microscope. All tumors are somewhat abnormal. That's how we know that they're tumors, but there's a spectrum of that goes from a poorly differentiated tumor, which basically means these cells aren't maintaining any of the normal features of what they should do, like appendiceal tissue should start to form glands and be organized in layers. Like a poorly differentiated tumor doesn't do any of that. Plus, on top of that, there will be like the nucleus will look abnormal. Like, for example, the signet ring feature basically means that the nucleus looks like a ring, which it should not. And so that's what grade is, and that's very important in appendix cancer, because the difference in outcome, in terms of survival for a high grade tumor is years different than a low grade tumor. So a low grade tumor, the median survival could be as long as 7 or 8 years from time of diagnosis, where it's more like 3 or 4 years for a high grade tumor, and moderate, which is grade 2, is in between. What's even more complicated than that is that within one patient.
Hmm. Hi, can anyone hear me?
Yeah, I can hear you, Amanda. You're still back.
I'm not
Yeah,
sure what happened with Dr. Shen. Oh, there he comes.
here he's back.
Ooh, sorry, we're back. Sorry, I had a moment of panic when that cut out. So where did it cut out with?
high grade.
Okay, got it. So yeah, so the grading is complicated, but that is if you ask one question about appendix cancer, first question is what is the grade because that affects prognosis and it also affects the treatment. So high grade tumors generally respond reasonably well to colon cancer style chemotherapy, and so this eventually we hope to find appendix cancer specific treatments, but until then, high grade tumors generally you're going to get colon cancer style chemotherapy, whereas low grade tumors, the colon cancer style chemotherapy doesn't really do a whole lot. We did a study that showed that 5FU, which is the most commonly used colon cancer chemotherapy drug, really did nothing at all for patients with low grade, grade 1 mucinous appendix tumors. So we don't give that to them anymore. So that's why the grade is so important, because it affects not only the outcome, how long you can expect to live, but also the treatment. With low-grade tumors generally being managed by surgery, not colon cancer chemotherapy. There's a lot of people out there that traditionally would say low-grade tumors don't respond to any chemotherapy, but I would push back against that. The traditional colon cancer chemotherapy has not worked for them, but likely there are other things out there that will work, which is something that we're actually working on with our clinical trials. We've had some success with immune therapy in these patients, and there's a recent paper from Dr. Lowy's group at UCSD showing the breast cancer drug palbociclib works in these patients. So the colon cancer style chemotherapy does not work well in low grade tumors, but I'm confident that there are drugs out there that will. We just need to find them.
What about promising clinical trials?
Right. So, I'll plug our own clinical trial. So we have a trial. Beth Helmick, who is a medical surgeon, is the principal investigator of it. Where we're taking paclitaxel, which is an old drug used in breast cancer, gastric cancer, esophageal cancer, and instead of putting it into the veins where it goes throughout your body, we put it into the peritoneal space, so directly into the abdomen, which, for most appendix cancer patients, the tumor is only in the peritoneal space. And so most of our ideas are fairly simple ideas. We think that if we put the chemotherapy where the tumor is located, we can get a greater dose to the tumor without getting so much dose to the rest of the body. And so we get more of what we call a therapeutic window, which is, kind of we need a dose that kills a tumor, but that doesn't kill too many of the normal cells, because that's where the side effects come from. And so that is in a phase 1 stage right now. Phase 1 means that we're still trying to optimize the dose. And so you have to kind of watch the patients very closely for side effects. So we can only treat 3 patients. And then we have to watch them for 3 months before we can treat the next 3 patients. Eventually, we'll move into phase 2, and so we'll be able to treat more patients on that study. But that has looked very promising so far. I know Dr. Foote, Mike Foote at MSK, Memorial Sloan Kettering in New York, is hopefully going to soon be opening a trial of BOT/BAL, which is bosilitamab. It's a combination from the company Agenus that has looked very good in colon cancer, just a very, I think, active immune therapy combination that is not open yet, but hopefully, in the next 6 months, that will open. And then we are working to try to open a KRAS inhibitor study specifically for appendix cancer. This is, still it's not written yet, so this is still kind of early days, but there are many KRAS inhibitor studies that are pan-cancer. So usually a phase 1 study will not be restricted to just one disease type. So you could, have appendix cancer, pancreas cancer, lung cancer, breast cancer, all on the same study if they have that KRAS mutation. And so we've put, I personally put, I think now 4 patients onto KRAS inhibitors. And for the most part, we've seen response. A few of the patients, it's very early, so we don't know yet how well it's going to work. But we know we presented this data in Barcelona last year at the ESMO conference, but based in our mouse models and cell line models, these drugs look very effective. And so we think that they will be effective in patients also. Obviously, we still need to to do the testing. But I encourage any, if you have access to a KRAS inhibitor trial and you have a KRAS mutant tumor, that seems like a great option.
So how about new systemic treatment options for treating appendix cancer?
Yeah, so the KRAS inhibitor I think is a really promising area. Um, that like most if you look at most solid tumors, any regimen that really is curative involves not just one drug but many drugs. And this is because the within the tumor in any one patient, not all the tumor cells are the same. And if you hit it with only one drug, there's always going to be some cells that are going to be resistant to that drug. And so probably if this is the case, like as with other cancers, the KRAS inhibitors are going to be effective, but to really to increase the durability of that effectiveness, we're probably going to have to do combinations. And so this is something that we're already starting to think about in the lab, doing testing what could be combinations of KRAS inhibitor with other partner drugs to really kind of increase both the fraction of patients who respond, and for the patients who do respond, increasing the durability of that response. And so I think that's maybe the most exciting area in appendix cancer. And then closely paired with that is better immune therapies. There's actually a lot of reason to think that combining KRAS inhibitors with T cell therapies, these are the drugs that you see advertised on TV, that the drugs like Yervoy, which is a CTLA-4 inhibitor, or the PD-1 inhibitors, of which there are many. These have not worked incredibly well in colon cancer, and part of that's a big part of why people haven't really tried them in appendix cancer. We did an initial study of atezolizumab, which is a PD-L1 inhibitor, and bevacizumab or Avastin, which was effective, especially in low-grade patients. But we think that adding in a KRAS inhibitor might make that combination work better. And we're also testing this with other kind of newer immune drugs and also newer VEGF inhibitors. So we think that this idea of immune checkpoint inhibition, basically activating the T cells combined with targeting the microenvironment with VEGF inhibitor, we think that's going to have traction in appendix cancer, and it potentially could be combined with KRAS inhibitor.
So can I guess the answer to this one? I'm going to say
Please,
data.
yeah.
Data. The biggest challenge in developing targeted therapies, data, patients.
Well, there's a lot of challenges. Lack of data is definitely one of them. I think that's one that we can definitely address and are addressing. Previously, lack of preclinical models. So like mouse models and organoid models was a big problem. That was actually in the first 5 years of our lab at MD Anderson, that was our number one focus, was to create these preclinical models. And so we actually just published a paper on this, but we've generated at MD Anderson, we've generated 5 what are called patient-derived xenograft, which basically means you take, used to be me, would go to the operating room, pick up the tumor from the surgeons and implant it in the mouse. Now we've got a team of a couple of scientists that do that. But we basically take the tumor straight from the OR, chop it up, and implant it into mice. And the tumor grows in the mice, and now we have a way to test drugs, before we go into people. And so the trial that I mentioned earlier, the intraperitoneal paclitaxel trial, that came directly from our mouse experiments. We did essentially the same experiment in the mice, and it worked very well. And that was kind of how we had the justification to do the trial in humans. And so we similarly, we're in the process of publishing our mouse data on KRAS inhibitors, which is providing the rationale and hopefully convincing the pharmaceutical companies to that this is going to work and it's worth them investing in a clinical trial to do this, because unfortunately clinical trials are very expensive, and so if you don't have convincing data that it's likely to work, the the companies that have the means to fund these are not necessarily going to do that. So that's been a big issue. Um, on top of that, some of the other problems is that because appendix cancer spreads primarily to the peritoneal space, it's hard to measure the effect of the drugs. And so you know, the if you're doing a clinical trial, the kind of most important endpoint is called overall survival, which is how long, if you could show that, people that get drug X lived longer than people that didn't give drug X, the FDA is going to approve your drug. The problem is that, appendix cancer patients fortunately live for many years. And so if you want to do a study where your endpoint is overall survival, your study is going to take 7 or 8 years to finish. And if you need to monitor the patients on your study for 7 or 8 years, that gets, very, very expensive. And you're not going to have your result until 8 years from now. So because of that, we try to find what's called a surrogate endpoint that we're confident that if we improve this, 7 years later, the patients will live longer. But that is challenging in appendix cancer because normally the surrogate is the tumor got smaller on a CT scan. And this audience probably knows that it's very challenging to accurately measure the size of appendix tumors on CT scans, partially because they don't form in like a ball of tumor, they form like a sheet. And on top of that, you've got a lot of mucin mixed in with the tumor cell. And so if you kill the cell part, that's what you want to kill, and the mucin is still there, it may not necessarily look that much smaller on the scan because the mucinous component is there, which is a problem. And so one of the ways we're working to try to combat that, we're trying to see if dropping the serum tumor markers like CEA, CA19-9, CA125 to see if that would be a decent surrogate. Basically, if we could say, okay, if the tumor markers came back to normal, we can call that response. One of the things that we're doing to kind of help this is looking at historical data to see, when people got chemo and their tumor markers went to normal, did they actually live longer? So if we could show that, we could now say our endpoint of this study is we're going to look at your tumor markers, 12 weeks after we start, and if they get to normal, we call that a response. Now we only have to watch patients for 12 weeks, which makes the study, one, makes the study much cheaper, and two, gets us an answer, years earlier. And that way we kind of get the answer from this so we can go on to, testing the many combinations that we want to test. So that, I think those are, there's many challenges to drug development in general, but I think those are some of them that are very specific to appendix cancer.
So, have you done or read any research regarding the effectiveness of that or that? I'm not going to try
Yeah,
to.
so I get asked this question a huge, I see patients every Thursday. At least one person asks me this every week. And it's there is a lot of data out there, and a lot of people really are promoting the use of these drugs. Unfortunately, there is some data in animal models that ivermectin can basically activate the immune system, but that's not specific to appendix cancer. And as far as I'm aware of, there's not any human data where people have shown that patients taking ivermectin do better than those that don't. There are some reports of people who say they've taken ivermectin and had a great result, but we don't. Lots of people are taking ivermectin. Whether or not their oncologists know about it, lots of people are taking it. So we don't know how many people took it and didn't get a good response, to know what the fraction of responders is. And so, it's not the worst idea in terms of, supplement, but, I think it is really, to answer these questions, it really needs to get studied systematically, and it just hasn't happened yet.
Okay, so how can the community do more to increase the number of CRAS trials that are done for CRC to include appendix cancer? I don't know if you covered any of this, but I guess the same question this person has is for all tumor agnostic trials that include a mutation seen in appendix cancer.
Yeah, this is That's a great, I mean, this is something that this is something that I try to do if I'm involved with the clinical trial at an early enough stage that it's still possible to modify it. I will always try to see if we can include appendix cancer. The difficulty is that from a from a research perspective, you're adding kind of heterogeneity to the cohort if you do that. And so if you don't because appendix cancer is very different than colon cancer, if you just sprinkle in a few appendix cancer patients, it's going to make the colon cancer data kind of harder to interpret. And so it would really be better to have a subset, that specifically, like 6 patients that are just appendix cancer that we're going to analyze separately from the colon cancer. But the other issue that we run into in terms of where we struggle to get appendix cancer patients onto these studies is that some studies will just have a blanket exclusion for peritoneal metastases. This is because there was one patient who had, I think it was with appendiceal cancer that had extensive peritoneal metastases that unfortunately suffered a bowel perforation. Because if the tumor is kind of grown all the way through the intestine, like full thickness through the intestine, and then you kill that tumor with the drug, which is what you want to do, that can leave a hole in the intestine where the tumor was, which is unfortunately leads to a perforation, meaning the intestinal contents get out, which leads to very serious infection, and potentially death. And so that which is a problem. The only way to really prevent that is to try to catch patients before they get really extensive intestinal involvement with the tumor. But that's been the other reason that it's been challenging to get these patients onto clinical trials because from a as a physician that treats appendiceal cancer patients, I push to try to put patients onto these studies, but I understand from the investigator standpoint, if you only have 16 patients on your clinical trial and one of them dies, that could shut the drug. There's not, if you have 2 patients out of 16 dying, your drug, that drug is probably never getting tested again. And obviously you haven't helped those 2 patients. So I see it from the other side now that we're running our own trial. Initially we tried to make the criteria as open as possible, but we actually had one patient who suffered a bowel perforation. So now we include patients with peritoneal metastasis, but if it's an extensive peritoneal metastasis that we think is full thickness, we now exclude them for that reason. So that's been part of the challenge there.
So, GNAS targeted trials, where are we with those?
So yeah, it's a great question. This is a major research interest of myself. We have a effort with some chemists out of UC San Diego to make chemical inhibitors of GNAS. Actually, full disclosure, we do hold a patent out of UC San Diego on chemical inhibitors of GNAS that we're trying to make the next generation of. That's very early in the drug development stage. Kirsta Flatmark, who is a surgeon scientist out of the University of Oslo, has a vaccine for GNAS that they've. I'm not sure if they've done their first clinical. I'm not sure if their first clinical trial is published, but I think they're. If it hasn't started, I think it's very soon to start, which is basically basically you're trying to get the body to recognize the mutant GNAS as being foreign so that it will be attacked basically by creating a vaccine, which is just putting the mutant GNAS peptide with some other things to stimulate the immune system. And so I know that is close to trial, if not in human trial in Europe. The other kind of promising therapy there, GNAS tumors tend to be mucinous. So we've shown, and other people have shown this, that if you turn the GNAS mutation on in a tumor, the amount of mucin production goes way up. There's a group out of Australia led by this guy, David Morris, who's a physician. basically started a company, and they have a, drug called Bromac, which is bromelain and acetylcysteine. Bromelain is actually like it's an enzyme extracted from pineapple. And then acetylcysteine is a standard chemical that is a reducing agent. It's also used to protect your kidneys from IV contrast sometimes, but it's very, very safe. That mixed together seems to be pretty good at basically liquefying the mucin so you can suck it out with the needle. And so that we've treated, I think, just one patient, MD Anderson, with Bromac. There's a group at UPMC that's doing this, and I think Trevor Groves at Mayo as well. It's been they've had a lot of issues with manufacturing. It's a natural products. I think they actually have like a huge pineapple farm in Australia, and they like have to like smash up the pineapples and then extract the bromelain. But because of that, the FDA has had issues with their batch to batch regulators. Basically, you got to like make 10 batches of it and send it to the FDA, and they all have to basically be exactly the same before they give you the thumbs up that you can, put this into humans. And so they've had some struggle with that. So unfortunately, like the whole, there's meant to be a phase 1 study of that that's supposed to open at Wake Forest, MD Anderson, Mayo, UPMC, but it's on hold everywhere pending supply. But we're pretty hopeful about Bromac in these mucinous tumors, most of which are GNAS mutant.
Interesting. Um, so, research on appendix cancer, how can it be expanded, and what role do patients play in advancing scientific understanding?
Well, I mean, one a simple answer obviously is funding. So the more funding we have, the more people we can hire, and so the faster we can kind of get to where we're going. The ACPMP has been very generous and funded our lab twice, which has been really tremendous. We are working on getting getting more federal funding. Right now, a lot of our lab is funded by the state of Texas. But having advocacy helps. A big part of why there is so much research funding for breast cancer is because one, it's common, but also because there's so much advocacy for it. Actually, a lot more people die of lung cancer than do breast cancer. But if you look at research dollars from the government, there's a lot more research dollars in breast cancer, and then that's largely because of advocacy, and people, tell their congressmen and their senators, hey, you need to support breast cancer research. And so I think there definitely is a role for advocacy in, just building out recognition that appendix cancer is its own cancer. It's not the same as colon cancer, and that we need to study it.
And to that point, like I have templates that you can write to your congressman, specifically around appendix cancer. If anyone wants to go to the Appendicure site, you can find them there, or just reach out to me. I can send it to you.
Yeah, and then it also related to that, not to, I don't want to make this sound like a plan for xCure, but again, sharing data online is really helpful because this is a rare disease. If we could have every additional patient that we get in our research cohorts is valuable to help us understand the heterogeneity from one patient to the next.
Yeah, and the thing is I do want to say this is like Wake Forest, UPMC, MSK. I'll go down the list of these great hospitals that are tackling this disease, but they can't sit down and share all their data, right? It has to go through specific software so that it can be made sense of. So I guess wanted to point that out in maybe layman's terms. Back to you, Dr. Shen.
Yeah, so I mean, we collaborate with the team at Wake Forest and with Memorial Sloan Kettering, also U Chicago, but we can't just like pool all the patient data, which is important because unless you the patient consent specifically that the data can be shared, we can't share it. So we could share it within MD Anderson, but we couldn't share it outside of MD Anderson. Because the way that we collect the data, there's not, we have what's called a waiver of informed consent. So basically, if you've been seen at MD Anderson, we have a protocol that allows us to, in an anonymous way, aggregate your data so we can learn from all of our patients. Because that really doesn't have risk to any one patient, we don't have to get informed consent, but to actually share data across institutions, that would require a consent. So we do like, so we can, like, for example, we have a study with Dr. Foote out of MSK where we're looking at, the benefit, or really the lack of benefit of adjuvant chemotherapy in stage 2 and stage 3 cancer. So basically the way that works is we put our data together at MD Anderson, and then they similarly do it at MSK, and we see, do we see the same kind of trends in our patients and their patients? And if the answer is yes, then we think it's going to kind of generalize. But it what it doesn't allow us to do is to kind of pull them all together to get a bigger pot to analyze as a group, which is something that the xCure platform can offer.
That's what I was trying to say. Thank you. So I guess the best, we all know appendix cancer doesn't really show up on CT scans and such. So what do you recommend?
Yeah, so we know that the scans tend to underestimate disease, but it's still the best thing we have. So high quality CT scan is what our surgeons at MD Anderson use. I know that some centers prefer MRI, but our surgeons and radiologists here are really operate off CT, and so that's kind of what I'm used to. Other places might use MRI, but so that's kind of number one. The tumor markers we find are helpful. What the tumor looks like under the microscope, the pathology, the grade is really critically important. So we treat, most of the, if you know one thing, it's what does it look like on the microscope? That's the grade. Number two is, where is it located anatomically? That's CT scan. That's going to be the main drivers. After that, the tumor markers can be helpful. Especially when, the tumor markers are especially helpful when one of the things is unclear. Like if it's not clear from the CT scan, is this getting bigger or smaller? That's where the tumor markers can be really helpful.
Okay, we have like 4 minutes left. I'm so sorry. We're not going to get through all these, but how can patients advocate for themselves when it comes to surveillance and treatment decisions? And I know from being in groups around all of this that a lot of times doctors, physicians, it's difficult for people to say, I would like other options, or what are the other options?
Yeah, so I think that's always a good question. If we didn't do this, like what would be the alternative? That's always a good question. I think it's fair to ask, what can I expect reasonably? Like, is this going to cure my tumor? Is it going to, just keep me alive longer? So like what can I expect? I think is a good question to ask. I think it's also fair to ask your treatment team if they don't, how many patients with appendix cancer do they see? And if the answer is not a lot, you may want to see a specialist that has more appendix cancer specific knowledge. But I think it's important, asking questions is important just because your doctor has a lot of knowledge about chemotherapy, etc., but you have more knowledge about your specific situation. And so I think that asking these kind of questions like what, if I do this, what can I expect? If I don't do this, what can I expect? Will help you kind of really make a more informed decision.
Yeah, you don't ask, you don't receive. So how much scar tissue is too much for a second surgery?
Yeah, so that's a great, it really like many things, the answer kind of depends. The decision whether or not surgery is a good idea is very complex. I think one of the other questions that we may not get to was, like one surgeon is offering me surgery, and others not. Why is that? And it's challenging. I'm a medical oncologist, so I'm a chemotherapy guy, so I don't absolutely make that decision. But it depends on, it really depends on what the surgeon think is going to happen. Like, surgeons don't become surgeons by not wanting to do surgery. However, they don't want to do a surgery that's not going to help the patient. And so if, like our surgeons, like they think that if we do this surgery and we do it right, and this tumor is going to come back in less than a year, probably don't want to do that because it's going to take a while to recover from the surgery. And, if we do all that and it comes back anyway, we don't really think we've helped you. Right. And so that's, um, but you could, I mean, it's hard to predict, is that tumor going to come back or not? And this is where we're trying to learn more with the biomarkers and molecular features. The grade is really important. Our surgeons are much more likely to operate on a low grade patient because those tumors grow slowly, much less likely to come back quickly versus a high grade tumor. So this is kind of evolving, but that's why that decision is complex and it changes. The one that, if you've had a prior CRS HIPEC, there's going to be a lot of scarring. And so that makes, normally you can do a laparoscopic, like with a small incision and a camera surgery to evaluate the abdomen. But if you've got a lot of scarring, you probably can't do the surgery laparoscopically. Because with all that scarring, with just a little graspers, you're not able to kind of take it all down. So the scar tissue usually is not going to prevent like an open surgery, like where you're going to try to take out all the tumor, but it definitely could prevent, and most of the time will prevent doing the surgery laparoscopically, to kind of just assess the amount of tumor.
Okay, so we are at time. I hate it, but thanks to everyone who's attended. Thank you, Tim. Thank you, Dr. Shen. Please reach out to me with questions that you've had as a result of some of the questions that Dr. Shen's answered. I'm going to send this recording out to everyone who registered, whether they attended or not, as well as the list of questions that everyone submitted with Dr. Shen's answers. So keep a lookout for that, and please reach out to me directly if you guys have any questions. Thank you, and have a great weekend.
Thank you all. And thank you, Amanda.
Take care. Have a great weekend.
Bye, guys.
Well, man, is everyone still on, or is it?
Yeah, they're
Okay, sure.
yeah.
Cool. Awesome. All right. Well, thank you so much for organizing.
Yeah, no, thank you so much. I mean, really, like that's what I said. If you don't ask, you don't receive. So maybe we can do this again. I think we're gonna
Absolutely.
get it. I mean, it was really beneficial to both of us. So I imagine a lot of people got a lot out of this. So thank you.
Okay. Yeah. Awesome. Take care.